Hi everyone, I'm Rike. I'm a PhD student at the Quantitative Biology Center, and I will talk today about sarek, so to catch everyone up that didn't know about it earlier. And we were just talking about it before the talk. Sarek is actually named after a Swedish national park, and it predates NF-core, which is why it got to keep its name and didn't have to get a descriptive name like every other pipeline. The pipeline is used for germline and somatic variant calling. Just to briefly catch you up here, so basically you have samples, tissue samples or blood samples, they get sequenced, in our case with short read technologies. Then the reads get aligned to a reference genome, and then we are interested in the differences between our reads and the reference, and we can do this for the tumor sample or the normal sample, or also look at the difference between both. And sarek does this basically in three big blocks. The first one is the pre-processing, which is based on the GTK best practices. We have the alignment step, so aligning the reads to the reference, currently with BWA, BWMM2, or DragMap. Then we do duplicate marking and base quality score recalibration, run a bunch of QC tools, and then have converted our input FASTQ files to CRAM files. And then the next big block is the variant calling. So here we have three tracks. The blue one that you see is for the normal samples. The red one has tools for tumor-only analysis, and then the blue and red track for paired calling. And you can see we have a bunch of different tools implemented at the moment for SNP and indel calling, structural variant and larger copy number calling, and microsatellites. And you can mix and match these tools however you need. You can run a bunch of different SNP callers all in parallel, or just one, or one SNP caller, multiple structural variant callers. And however you need for your analysis. And then the last big block is the annotation part, so assigning knowledge and also predicting protein effects and so on. As you can see, we have several of these entry points, so these file icons here, to facilitate easier reanalysis. Let's say a project runs for a long time, and you were initially supposed to do some sort of SNP calling, and then throughout the project somebody also would like to do structure variant calling. Then you don't have to do this really computationally heavy pre-processing, but can just start from the variant calling, and you don't need to keep the work storage around the whole time. And also we have round and then little squared station in this metro map overview, and the round stations are all optional and only the squared stations are mandatory. So sarek is essentially just a huge wrapper around BWA, and you can turn off tools or turn them on to tailor it to your needs. The pipeline supports whole genome, whole exome, and panel data analysis, and as for short, paired, and reads. The pipeline was originally designed for human and mouse samples, and with that we just mean now that basically for those organisms we ship all the reference genomes already, but it's been used for other organisms like pigs or tomatoes or bees or so, and you at least need a FASTA file for this, and then maybe need to provide some of the annotation files or so manually. Right, so Maxime already mentioned that sarek was started a long time ago, 7 years ago, and several institutes in Sweden. Was ported to NF-core in 2019, and then I somehow joined the team and showed up, and we started porting sarek to DSL2, and then released the first version of it last year. And while we were porting sarek, we figured, since we have to rewrite the whole pipeline from scratch anyway, touch every bit of code, we should try and optimize it here and there a little bit. Also mostly motivated by some of my own research projects that didn't quite work out because of some bottlenecks. Yeah, and at the moment, sarek is mostly supported by people from QBiC, the CRAM Labs, the Danish National Genome Center, and many other volunteers in the community, of course. Right, so one of the major or main things people complain about is that sarek uses too much storage space. So the work directory gets too big, it's a bottleneck, and it's then hard to use. So we figured, why not try using CRAM file? It was on the roadmap for a long time, and there was always something preventing it. Not enough tools were supporting it, or there were other issues. But then 3 years ago now, actually, we tested all the tools, and it worked fine. So that's what we did. We converted the whole pipeline and then used CRAM files everywhere. And it happened, like the thing we expected actually happened. The work storage got massively smaller when we took the same pipeline with the BAM and the CRAM implementation. Also a bit faster. We did use more memory for some of these processes. In practice, this wasn't actually an issue because it was minimal per process, and then memory, of course, can be reused by further processes down the road, but the work storage just keeps accumulating. Right, and then the other thing that we looked at is how does one sample travel through the pipeline? Nextflow, of course, parallelizes across samples, but what does one sample do? And we start from the FASTQ file, and what we then added is that for each FASTQ file that enters into the pipeline, we'll send through a tool called FastP that splits the sometimes really big FASTQ input files into tiny baby FASTQ files, and then we run the alignment step in parallel. And this made the whole alignment step much, much faster. We did double the storage space for the FASTQ files because they had to be written once to the disk, but it was still worth it. And then duplicate marking takes care of the merging, and it didn't have any further time punishments or so on. And then the other thing we investigated is the big step-based quality score recalibration and variant calling. So GATK recommends a scatter-gathering approach where you parallelize across genomic regions. So it can be multiple chromosomes or even parts of chromosomes. And especially for the human reference that we are using, there are about 370 regions specified. And sarek kind of tries to be smart and groups these regions together to process them in multiple logs or groups. And we just had some number that has always been there, some type of grouping, and then we figured we should investigate. Is this actually a good grouping? Should we maybe do it a bit differently? And turns out we really should have done it a bit differently. Yeah, so let me show you the results here. So here I again ran the comparison on 10 samples. On the left you can see the runtime always for the longest running group. And then on the right side, the accumulated work directory per sample, work directory space per sample. And in sarek 2 and very early version of sarek 3, we for some reason just had about 124 interval groups. It was just the case. And the whole point of the scatter-gathering approach was to speed up the analysis, but as we can see, we don't actually speed up much further, or we don't get much faster after 21 intervals, but for some reason the work directory just keeps on growing massively. So we scaled this down. So now with the new default, it's 21 interval groups. And this again then reduced the work storage by about a factor of 4, and then also reduced other computational resources. Right, and then one of the last things we were interested in how do the cloud costs, or did the cloud costs develop over these different implementations? Here we checked it on AWS and US East using spot prices, the EBS autoscale, and then the old fusion mount, so none of the fancy new things that Paolo showed today. And in sarek 2, we ended up with about $70 per sample, quite expensive. Then in sarek 3, we reduced it to 25. And then when we reduced the number of intervals further, we are now down to about $20 per sample. And I'm quite curious to see what all of these memcloud and new fusion things and so on will bring. Also, the pipeline got faster. We also fixed a bunch of things where we had accidental bottlenecks in the pipelines and so on. Okay, and then before I come to the end of my talk, I just wanted to highlight some of the other cool new features that were implemented this year. So Anders spent a lot of time adding Sentieon support for mapping, duplicate marking, and I think as of this week, various variant callers. So it's a license-based commercial tool that can be used for these steps that is supposed to be much, much faster. Not only did he add it to sarek, but he managed to add it to NF-core modules by getting a test license so it can be used by other pipelines, also without the developers needing the license themselves. QBiC here added joint calling for MUTEC 2. So when you have multiple tumor samples, you can now do joint calling. Maxime, of course, added the annotation cache that he talked about earlier, as well as an option to only build the indices. So you can pre-run sarek before you do anything, build the indices, and then share them, for example, at your institute if you run on one of these organisms that are not currently in iGenomes. And then the last thing we looked more into is benchmarking with genome-in-a-bottle data, looking at Illumina reads, of course, but also other technology together with Lasse and Edmund. The results look more or less as expected, and we are trying to now automize this on every release. So I got to rope in Edmund this week to help me with some issues, and we basically want to push the VCF files and then compute these F1 scores automatically and display them, because I could do it manually on release, but I know I won't and I will forget. And we should really have this on every release to make sure everything works as intended. I'm at the end of my talk, and I also want to use the opportunity not just to thank the usual people, but especially everyone who keeps contributing, who keeps answering issues, and just helping us making sarek more awesome and better and fixing everything. Thank you so much.
All right, awesome work. Well done to you and the whole team. We have a few questions, and we should talk about cost optimizations, by the way. A few questions. One, is it possible in sarek to merge BAMs but turn off duplicate marking?
To turn off duplicate marking? Yes.
Yeah.
Yes, then SAMtools will just merge all the BAMs, convert them. SAMtools will merge everything then, and you can circumvent it, the duplicate marking, yeah.
Nice. A kind of a bigger scope on the second question. What are your recommendations for developers of other pipelines to increase speed and decrease resource usage, especially for developers that start from scratch?
Ooh.
It's a big one.
Bring a lot of time, I think. That's at least what I did. I think one of the most surprising things for me is, or that I found out, is that just cutting things up and parallelizing more and like running things more in parallel is not always better. So, and we try with small data sets. I tried with big ones in the beginning, and I wasted a huge amount of time on this. So,
yeah, I would echo that. It's definitely worth taking a little bit of time to see like how you chunk things up, depending on what kind of infrastructure you're running on as well. It's worth doing that and benchmarking, because it can make huge cost savings if you're on cloud, and speeds, savings, etc. Anyway, sorry, this is a topic near and dear to my heart, so don't let me start talking. It's not what I'm here for. Okay, well, thank you very much. I think that's the question. Oh, wait, sorry. Oh, there's actually a lot more. One more, and then we'll go to the next talk. How can we help new developers to work on a massive pipeline like sarek?
Come and ask us lots and lots of questions. I think we all really want to help, and sometimes maybe we are not fast in replying because other things happening, but please keep asking. I really don't mind being reminded 5 times to help, and I don't know, we get on Zoom calls, we help each other out, and yeah.
All right, thank you very much.